Really happy today to have Kagan Karakus joining us. He's an MD and he is based in Aaron Michael's lab at the Barbara Davis Center for Diabetes at University of Colorado School of Medicine. The title of his talk is T-cell receptor sequences targeting B-cell, beta cell proteins, are present in blood across the stages of T1D or type 1 diabetes. And just a short bio on him. He graduated from Koch University School of Medicine and is currently a postdoctoral research fellow, as I mentioned, in the Michaels Lab at University of Colorado. His translational research focuses on T-cell receptors as potential biomarkers in type 1 diabetes. And he recently received an award from the ADA, or two awards from the ADA. He received the Andrew Goldberg Early Career Award, as well as the Abstract Award for the abstract he's going to present here. We're really pleased to have him, and we ask that if you have questions, drop them in the chat, and he will take them at the end. I also have the link to his abstract in the chat. And great to see you, Kagan. We're really excited about this work coming from the Michaels Lab and from your efforts. It's really, it looks as if these have some real potential for biomarker use. So we look forward to hearing the talk. Thanks for coming.
Thank you. Thank you very much. And it's so good to be here. And I will be presenting about T-cell receptors, as you said. But I will be presenting two projects. And I can start now. I don't have any disclosures. First, I want to start talking about T-cell receptor beta chain sequencing. So as you may know, T-cell receptors has two chains, alpha and beta, and here we are only sequencing this beta chain. It has three important high variable parts, CDR1, 2, and 3. And this CDR3 is important for epitope recognition. And also the other CDR parts are important for the MHC interaction. And we are only sequencing this beta chain because it has some advantages when we only sequence this one. First of all, it provides high depth and sensitivity because we can reach millions of reads and also almost a million T-cell receptors. And this allows us to detect very rare clonotypes in a large repertoire. And also, it has very simple sample requirements. We can work with bulk DNA, and we don't need viable cells. And this molecular assay makes the life easier. And it's scalable, so it allows processing many samples at a time, so that we can run multiple samples. And also, it's good for diversity and clonality analysis with a high number of T-cell receptors detected. It's robust for repertoire profiling, and this also allows us to do some monitoring over immune system over time. And relatively, it's cheaper with this lower cost per sample. It's suitable for large-scale studies and longitudinal tracking. And this is relatively an older and well-established method, so there are established protocols. And previous efforts has been done in sequencing this T-cell receptor beta chains, and we can identify these antigen-specific sequences. In previous publications, we see that these beta chains can be used in cytomegalovirus or SARS-CoV-2 virus and other immunity tracking for viral peptides. And also our lab has shown we can use this T-cell receptor beta chains to track this islet antigen specific T-cell receptors in type 1 diabetes. But why? Why are we sequencing this beta chain, and why are we sequencing T-cell receptors? Why is this important? So as you know, type 1 diabetes is a progressive condition, and this progressive condition has definitely some uncertainties, and we cannot detect or predict the timing of progression. That's why we are aiming to detect and use this disease-relevant T-cell receptors as a non-cellular biomarker during this type 1 diabetes development. So I can start with the first study, first project we did. So here we sequenced 920 samples, and of them 712 were samples of individuals with new onset type 1 diabetes, and we had 208 control samples. And these were pediatric groups. So as you can see, the mean age here is 12 for both groups. They were mostly non-Hispanic whites. And we collected all diabetes, type 1 diabetes samples within 100 days with a mean duration of 10.8 days, which is also important. And as expected, hemoglobin A1C is 12.2, which is quite high, and that's expected for a new onset group in a pediatric population. And when we look at the antibodies of this group and of these samples, we detected 4 major autoantibodies. They are prevalent as expected. There were only 8% of group with 0 autoantibodies, 14% had 1 autoantibody, and almost 80% had multiple autoantibodies. And these are in line with other studies and previously known autoantibody profile for new onset pediatric groups. When we look at the HLA, we've, of course, the high-risk HLA haplotypes, DR3DQ2 and DR4DQ8, they were both prevalent in diabetes group. They were not as prevalent as diabetes group in controls, as you can see here. So we sequenced all these 920 samples. We used their genomic DNA and we sequenced them using adaptive immunosec assay. And with this assay, we had the T cell receptor beta repertoire, and we covered entire repertoire. So we got a total sequences of 268 million sequences. And when we look at the distribution per sample, you can see here on this graph, and type 1 diabetes and control groups had very similar distributions. And we got an average of 290,000 T cell receptors or productive rearrangements here in this study. So next, I want to talk about what we did with these sequences we got. So first, we created a list of islet antigen-specific alpha and beta T cell receptor sequences from the literature. We searched PubMed and we found the publications since 2012, and we identified full-length and paired T cell receptor alpha and beta chain sequences in these papers, in these publications. And these publications determined this antigen specificity by using these 4 methods. And this is important because here we are using this reference list as the source of antigen specificity. And we searched our samples, we searched this curated list in our samples and to find exact match of these published known T cell receptor beta chain sequences. So let's have a quick look on our reference list. So we created T cell receptors for islet antigen targeting TCRs. And as you can see here on the left-hand side, most of the T cell receptors were targeting GAT and preproinsulin. And also we had some sequences targeting zinc transport rate, IGRP, islets, and IA2 as well. And also, we also created some T cell receptors targeting viral epitopes, viral antigens. Most of them were targeting influenza and EBV. We also had some CMV, adenovirus, and Coxsackie virus targeting TCRs here as well. And as I said, we searched for these known antigen-specific beta sequences, T cell receptor sequences in our 920 samples. And we found that this 31% of these known T cell receptors were shared in our sequences. Also, we found 52% of viral sequences in our samples. First, I want to show you the viral sequences. Here you are seeing the percent of samples with at least one T cell receptor for different viral viruses. So, for example, here in almost all samples, we had at least one influenza targeting T cell receptor as expected because it's a common virus. Almost everyone got exposed to these viruses in their daily lives, which is quite expected. And also, we found 80% in our samples, we found EBV targeting TCRs in 80% of our samples and 60% of CMV. So, and as you can see, controls and T1D groups didn't differ. They look quite similar as expected. Next, we look for diabetes-related T cell receptors, and we found 529 islet antigen targeting TCRs, and they were prevalent in both groups. Here, when you look at the distribution of unique T cell receptor sequences in each sample, these are very similar. And that's why we concluded that some of these TCR sequences are not specific for diabetes. That's why they look very similar. And this can be physiology or just benign autoimmunity. That's why we decided to first make some selection, and we selected the specific ones. I think this was the lowest hanging fruit there. So this, when I said this type 1 diabetes specific T cell receptor beta sequences, we have 156 of them, and they are present in at least one type 1 diabetes sample, and they are not present in any control samples. And with these samples, we can cover 42% of samples, and they were coming from 45% of the patients. And as you can see, they cover all age groups, so they are not specific for any age. They are coming from all. But still, it only covers like 42%. That's why we wanted to increase our coverage. So we decided to add some other TCRs as well. So as I said, we had 529 TCRs. I showed you the specific ones. And from the non-specific ones, we decided to filter them and test them if they increase our coverage in a good way. So we applied these filters. If a TCR is present in less than 4% of controls, and if it's more prevalent in diabetes samples, then we selected them and we reached 101 TCRs. Next, we tested them iteratively. We added them one by one and in combinations, in many combinations. And we selected these 8 TCRs here, and we reached a final number of 164 disease-relevant TCRs, so-called. And with this addition, we increased our coverage by 10%, and we kept our specificity is quite high, it's almost 95%, as you can see. That's why we picked some control samples here. And when we look at the profile of these T cell receptors, most of them were targeting GAT and PPI, and we had some zinc transport rate, islets, IGRP, and IA2 targeting TCRs here as well. And next, we wanted to validate this. So that's why we use longitudinal samples coming from DAISY study. DAISY study is a longitudinal birth cohort in high-risk individuals. They were followed from their birth until they developed diabetes autoantibodies and also afterwards. So when we look at the performance of these selected T cell receptors, they were present in 80% of cases, which is quite nice. And when we look at this, look at the graph here or the illustration. So you are seeing an individual on a row, and you can see the repeated measurements here on a row. And we highlighted islet antibody, autoantibody seroconversion here with this black box. And as you can see, these disease-relevant TCRs are predominant at or before islet autoantibody seroconversion. which is quite nice. However, we also found these TCRs in control samples, almost in half of the control samples. And after this, we actually we decided to design our next project in a different way because we found these TCRs. Only 1/3 of them were specific, but we have a very big group of non-specific TCRs, and there can be some TCRs they are important and maybe expanded in disease, but not expanded in control samples. That's why to detect them, we decided to design our next project in a targeted and deep T cell receptor beta chain sequencing. When I said targeted because we also realized that we can just use 1/3 of the V genes, and we can still cover almost 90% of diabetes related or type 1 diabetes islet antigen related T cell receptors by just using this 1/3 of the V genes. Now I want to also talk and introduce you this the method we used in the second project. We used a nested PCR for targeting these beta chains. First, with our primers, we selected this beta chain part from the genomic DNA. Next, we added the sample index for each sample, and we amplified this T cell receptor sequences. And after that, when it's ready, we use next generation sequencing in our samples, and we used our in-house bioinformatic pipeline to find this T cell receptor sequences in our samples. And with this time, we used 27.2 million reads per sample, which is much larger and greater than the previous project. Here we had 18 stage 1 samples, 24 stage 2 samples, 40 stage 3, and 40 genetically at risk autoantibody negative samples that we used as control here. And these are cross-sectionally studied samples, meaning that they are not longitudinal. However, they are well characterized for each stage here. And you can see the amino acid clonotypes or amino acid level T cell receptors here. We have a very even distribution between groups. So this shows that we had a similar depth of sequencing in our samples. And we also, since we are using genomic DNA, we can also detect nucleotide sequences for each T cell receptor as well. And when we look at the DNA clonotypes, these are nucleotide sequences, we are seeing 100 to 200,000 difference for each sample we have. As you can see, it's moved a little bit up here if you compare these samples. And now I want to introduce you this discrepancy between amino acid sequences and nucleotide sequences we have. So these different nucleotide sequences can still produce the exact same amino acid sequence, as you all know, as you may know. And this phenomenon known as convergent recombination. And I try to visualize this. So when we look at the first clone here, there are 4 cells, and they all have the exact same red T cell receptor here, shown in red. And these 4 TCRs have the exact same amino acid sequences here. However, there are actually 2 different DNA nucleotide sequences, but still they are producing the same T cell receptor. And when we look at the second clone here, they all have this same T cell receptor here, shown in blue. But you can see that there are at least 3 different T cell receptor nucleotide sequences still produces the same T cell receptor. So here we are hypothesizing this, that if we see more nucleotide sequences producing the same T cell receptor as amino acid sequence, then it's more likely that these clonotypes had expansion, clonal expansion. That's why we are seeing this diversity in nucleotide sequences for a given T cell receptor. That's why, as I said, we use this deep sequencing method so that we can detect these nucleotide sequences or DNA clonotypes that converges the same amino acid clonotype, and we use this as a clonal expansion. So I want to show you what we got once we sequence the sample. So as I showed you, we can detect EBV and influenza CMV. And here, as you can see, this is one TCR as an amino acid sequence. However, there are 10 DNA clonotypes producing the exact same amino acid. And we detect this also in GAT samples, GAT TCRs, preproinsulin TCRs, and also zinc transport rate TCRs as well. And we are not just detecting antigens, we can also find epitopes if they were stated in their original manuscript in the original study. For example, for these PPI samples, we can find C-peptide, hybrid insulin peptides, signal peptide A chain and B chain targeting T cell receptors. And they were mostly coming from CD4 cells, and I highlighted the T cell receptors coming from CD8 cells in a different color here. And it's also the same thing for GAT. We can find some GAT epitopes if they were stated in the original manuscript or in the reference manuscript. So what we found in our sample was that we found 276 diabetes T cell receptor sequences from the curated list, and they had similar frequencies between groups as seen here, and this resembles the data I showed you in the previous project, and we had the same conclusion. Some of these sequences are not specific for diabetes. And also, I want to show just one TCR here. So you are seeing the sequence of this TCR. It uses this V gene and J gene, and also it has this CDR3 region. And in the original manuscript, this TCR was targeting GAT65, and it was found in a CD4 cell. But you can see that it was present in many samples in all groups. And TCRs like this really reduces our specificity. That's why we had selection. We had to make some selection. Again, we selected the specific ones. See, for example, this TCR was present in 3 stage 1, 2 stage 2, and 3 stage 3 samples, and it was absent in control samples. And also this time, with this deeper sequencing, this allowed us to use this expanded TCRs, and we included these TCRs in our curated list. For example, this TCR, yes, this was present in 5 control samples. However, it was more prevalent in stage 1 and stage 3 samples. And more importantly, here one sample had 5 different nucleotide sequences produce this TCR. And we use this as a proxy for expansion, as I mentioned before, and we included these TCRs as well. So we selected 83 TCRs, and they performed much better as expected. So we can see definitely the important difference here for DNA clonotype frequency between control samples and also across the stages like stage 1, 2, and 3 had higher frequency than control samples here. And when we look at the profile of this group of TCRs, most of them were targeting GAT and preproinsulin. We also had 1 zinc transport rate targeting and 2 islets targeting TCRs in our list. So one may think if HLA is important in the performance of these TCRs, so that's why we analyzed this HLA-DR genotypes and the frequency of these selected TCRs. And as you can see, we didn't see any HLA restriction or any selective performance of a given HLA-DR group here. And also, we had we had the breakdown of this for GET TCRs and pre-proinsulin TCRs. And also, as you can see, it didn't differ between groups here as well. So we can say that this TCR doesn't perform selectively for a certain HLA-DR group or HLA type. Next, we looked and we searched for the interaction or the association between these TCR performances and autoantibodies. And you are seeing the number of positive autoantibodies on x-axis and the frequency of these selected TCRs on y-axis. We had a decent correlation between these two, and this was also expected. However, it's hard to say a cutoff or put a cutoff here to really distinguish and make this relationship binary. So we have a good correlation here, and we also break this down for GAT and PPI. And there was a good correlation between GET T cell receptors and GET antibody levels. And also, to a lesser extent, we found this correlation between PPI targeting T cell receptor sequences and insulin autoantibodies, insulin autoantibody levels, to be exact. And when we look at the progression or the profile of these T cell receptors across the stages, we noticed that stage 1 group had the highest frequency. However, there were only 33 T cell receptors out of 83 selected TCRs there. And in stage 2, this number increased to 40. And in stage 3, this increased to 53. And more importantly, in stage 1, we mostly saw GET targeting TCRs in this group. And in stage 2, we saw that there is some addition of this insulin targeting TCRs. And in stage 3, we see even a greater diversity of the epitopes that were targeted. So that's why this was really exciting to see this T cell epitope spreading across the type 1 diabetes stages. And next, we wanted to understand if age plays a role. That's why we focused on the stage 3 samples. And since the beginning, we designed this experiment with a certain age considerations. That's why we prepared this age bins from age 1 to 5, 5 to 10, 10 to 15, and 15 to 17. And there are 10 children in each bin. That's why there are equal number of children here. And as you can see, as the groups get older or older age, we find less T cell receptors, and also we see less diversity for adolescents here. And before age of 10, we can see many PPI targeting TCRs. However, for the adolescent group, we are almost only seeing this GET targeting TCRs, and they were coming from less number of unique TCRs. And this is, I think, very important, and that's why we wanted to investigate this further. So we looked at the relationship between age and this frequency of selected TCRs, and we found a significant correlation. It's not a strong one, but still, we found this correlation. As the groups get older, we see less frequency. And we also wanted to see if this is coming from PPI TCRs or if this is coming from GET TCRs. And the answer is it's coming from PPI. We see that significant correlation, and GET TCRs didn't have a correlation here. They were present across all ages with similar frequencies. However, when we look at this PPI T cell receptors, we found that in older ages, like in adolescents, actually, not older ages, but in this adolescent group, there is this absence of PPI targeting TCRs, and this absence of PPI TCRs actually drives this correlation. And we didn't see higher correlation in the younger or earlier ages, like less than 5. However, this absence looks important for adolescents. And still with this data, we have many things to do. So right now we are working on this T cell receptor clustering around this type 1 diabetes specific and expanded reference sequences. Also, by using this paired T cell receptor alpha and beta chain sequences, we can detect more specific antigen-specific T cell receptors. That's why we are planning to use turtle-seq for this. And also, deep sequencing of longitudinal samples can be beneficial, and we are planning to use this as a validation, and also it can gives us more insight about the development of diabetes throughout the stages. And also in a separate project, we are planning to measure this antigen-specific T cell receptor beta sequences pre and post immunotherapies like anti-CD3 monoclonal antibody. And to summarize, this T cell receptor sequencing I showed from genomic DNA can provide novel insights into diabetes progression and pathogenesis. And this targeted and deep sequencing may prove to be a robust biomarker of active autoimmunity in type 1 diabetes. And with that, I want to thank Dr. Aaron Michaels and all the lab members and also all the collaborators at Barbara Davis Center, and also all the funding agencies. And I can take the questions now. Thank you.
Thank you, Kagan. That was really fascinating, and there's a lot of work there to kind of just sort of tap hats off to the whole group, especially Aaron and you know, the whole Michael's lab. You guys are sort of following the T1D TCR expansion as or the epitope spreading, right, as the patients progress to T1D. Is there ever a time that you've noticed any kind of contraction, sort of to mirror a relapse remission phenotype in the prodrome? Like, do you ever see like a really, some kind of spread and then another window of decline?
Um, we need um, we need longitudinal samples for
Mhm.
that. Actually, that's one of our future directions. We are actually hoping to see such an expansion, or as you said, like some kind of shrink in the longitudinal samples. I think that will provide more insight about the
Yeah.
development and progression of diabetes.
What other resources could you potentially tap into to, gain more longitudinal windows or to that are already existing, or to actually just gain more samples outside of Daisy?
Um, Resources here at Barbara Davis Center. Actually, we have a great data bank. We have many samples. We will use our own. And also, there are other ongoing big, large studies with
Mhm.
longitudinal sampling. So they can provide good resources uh, like TEDdy or others. So TrialNet. But
So you guys have that access and you can just, incorporate these resources into your protocols that you've already developed.
Um, so I don't know that part. I think Dr. Michaels will probably uh tell
He's on here,
that part. Um,
Aaron.
from the
Yes, no, maybe.
now the account for NIAT.
What do you say? Okay, well, let's go to
I then
Peter.
mute myself. Yes,
Oh,
so we will yeah have a new grant that was very well scored to do sequencing in TEDdy samples, again longitudinally, and that's for both alpha and beta chain sequencing. So not not paired, but we 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 do very much want to do paired sequencing and try to up the numbers and work with
yeah.
Paul Thomas for that turtle-seq assay where we're getting up to 50 to 70,000 paired alpha beta T cell receptors from a PBMC sample, which is pretty good for 2025.
Yeah, that's awesome. Great news. That's exactly what I was kind of interested in understanding. So that's really exciting. From Peter Lindsay, for the relationships between TCR frequencies and insulin antibodies, how do you account for neo-autoantibodies resulting from insulin treatment in new onset type 1 diabetes? And what if you exclude TCRs directed against mature insulin?
So we used only stage 3 samples if they had diagnosis within 21 days. I think that addresses to this problem to some extent. And if you exclude TCRs directly as measure. And also we still saw C-peptide targeting TCRs there. And I think that's also gives us some hint about targeting the endogenous, but not the exogenous insulin. But yeah, we really try to address this problem by limiting the duration of sampling within 21 days of diagnosis for stage 3 samples. And stage 1 and 2 samples, those individuals didn't use any exogenous insulin. So
Great answer. We had a little talk offline, and I wanted to see, just to kind of highlight that, just sort of the volumes that are needed, that you guys are drawing out in terms of if thinking about future screening or, biomarker tracking, what kind of volumes, are you using now, and how might that translate to patient volumes in the future in terms of blood volumes, like, right? Because you want to be able to track those who might have these aberrant TCRs.
So we are using 2 milliliter of blood right now, which is a quite less compared to for the blood amounts we are using for other purposes. And this is a molecular assay, so we don't need viable cells, and it makes the storage of these samples or the resources in a much better way. So this is still in development, so this is not a commercial assay. That's why it's hard to say something about the scalability of this protocol, but it looks quite doable because it's PCR based. And also, and beyond that, actually, as I said, this is molecular assay, and it's not using viable cells. That's important.
And
And
you also, I think we also talked about the fact that you're using a very small volume of DNA.
yeah. Oh, so yeah, I remembered. Okay, I was going to address this screening part. I forget that in a moment. So actually, for screening, we had great tools like autoantibodies, and these TCRs can be more important for monitoring instead of screening, because antibodies still can't address or predict the time of progression to the next stage. Maybe these T cell receptors can be used as biomarker for progression and can be used for monitoring in that regard.
Yeah, and you've got, like, you're only using right now like 10 micrograms of DNA to pull that out. So that's also very nice because you don't need that much.
Yes. Yeah.
Great. Well, this is really exciting, and no surprise that you did win those awards at ADA. That's a lot of work, and it's really heading in some very exciting directions too. So congratulations, Kagan, and thanks for the talk. And we'll be looking for the paper coming out of that abstract and featuring in our week in review.
Yeah. Thank you. Thank you very much.
Thanks again. Have a great rest of your day.
Yeah. You too.
Bye.
Thank you very much.
Bye-bye.